So hello everyone, my name is Wolfgang Rankeln and I'm sitting here with Linda Amaral Zettler from the US, and today we're going to talk about the GSC14 and the topics. So maybe first of all introduce yourself and talk a little bit about your scientific fields that you are working on.
Sure. I'm Linda Amaral Zettler. I'm from the Marine Biological Laboratory in Woods Hole, Massachusetts, where I'm an associate scientist. I also teach at Brown University in the geology department. My background is in biological oceanography with an emphasis on molecular ecology and microbial diversity. And for the past several years, I've been intimately involved with the GSC through helping develop standards, specifically for marker genes. This is the MyMarks standard. And part of the interest in that grew out of my involvement with the International Census of Marine Microbes, where we really try to, in addition to collecting information about latitude, longitude, time, and depth, we were also very interested in collecting contextual information attached to the amplicon sequencing that we did as part of that effort. And I served as the program manager as that during 2004 and 2010 when the census was active.
So what would you say is the contribution that the GSC made to a broader community?
Well, I think it's, we've been sequencing DNA for many years now, and I think the value that the GSC brings to the community is the added value of contextualizing that kind of data, because more and more as we sequence more environments, the more we know about those environments, the more intelligent analyses we can accomplish. And I think for at least a period of time, people were, I don't want to say negligent, but they were reluctant to spend the effort and time into doing that. But I think with some of the recent publications that are coming out or are starting to come out that really can perform wonderful meta-analyses of these kinds of data, people are starting to appreciate the value added of this contextual data. And so I think the GSC provides a framework for reporting those data. The other huge, in my opinion, service that the GSC has done is to really has been to speak with the places like the INSDC and cooperate with them and work with them to be able to allow people to report this information alongside their molecular data, because that itself was a big change in the data repositories, was the acceptance of that.
So this is now the 14th meeting of the GSC. What were the topics that were particularly interesting to you?
Well, this meeting was mainly about place-based science, and the work that I've been doing has in part been place-based through a project I have that's been going on now since 2007 called MIRADA LTERs, which stands for Microbial Inventory Research Across Diverse Aquatic LTERs. LTER stands for Long-Term Ecological Research Sites. So these are sites where the National Science Foundation in the US has dedicated resources to long-term studies of ecology and other kinds of questions that need to be answered over longer timescales. And prior to my project, there hadn't been a lot of microbial diversity work done in some of the sites, not all of them, but some of them did have work that was being done. So my project was one of the largest cross-site analyses of the program. And in part, I was interested in GSC in being able to leverage the work that I've been doing and to be able to bring some of the concepts to that community as well. So I've been working in different levels in that capacity.
So maybe can you explain it in your own words? What is a genomic observatory?
Right. So when I hear genomic observatory, because I do a lot of diversity work mostly with single marker genes. When I hear genomic diversity, I think more in the context of genomes, as opposed to genes. So I think the genomic observatory brings a genetic study, a biodiversity study based on single marker genes to a different level, where you're considering the genomes that make up part of that environment. So I guess when I think of genomes, I think of metagenomes, and so not necessarily sequencing single genes, but a family of genes that can tell you something about the metabolic potential, the metabolic diversity. I am a bit of a traditionalist. I think of diversity in terms of genes that follow evolutionary lineages, such as ribosomal RNA. But in fact, there is a wealth of other kinds of diversity present in an environment in the form of other kinds of genes that serve other purposes. So not only is there richness in terms of species, but there's richness in terms of genes and their metabolic products.
So why do you think it's important to set up a genomic observatories network?
Well, I think the reason to set up a network is the same reason that, for example, I went forward and tried to do a cross-site study with a coordinated effort and, common protocols. The ability to ask big scale, high order level questions is really facilitated by common approaches and common data reporting and, just a standardization. Is important, and I think there are questions you can ask at that level that if you're not coordinated, you can't ask. And it's another is just a practical reason, and that is that there's limited resources, and so you shouldn't be reinventing the wheel if someone has already thought about a problem or an approach or even tested it. And so I think it's really important to leverage that knowledge and build on that knowledge as opposed to repeating it.
So what are these big scale questions that you would like to address with this initiative?
Interesting. Well, I personally, I think I'm part of a community of scientists that is very interested in better understanding, I think, the value of genomes that are present in the environment. Really what we don't know, what's left to discover. Myself and many colleagues are very much in still the age of discovery, and I think we as a community are still at that stage. And so I think it is important to better understand the genomic resource that's available in our communities, in our oceans, on land, anywhere. I think it's important to do. I think there's still a lot sort of remedies and resources that we can derive from the sea and even the land. Antibiotics, there's all kinds of resources, I think, that we at least need to be aware of. I'm not saying we should exploit them, but I think we need to be aware of them because they can benefit us.
Okay, you already mentioned that there's already some infrastructure out there, and we shouldn't reinvent the wheel. So what are the plans to integrate this initiative into the already existing infrastructures?
Well, that's part of what we're discussing at the meeting. Hopefully we'll come up with a strategy that will work and that will bring people together as opposed to splinter them. It's always challenging organizing and motivating individual groups to come together. There's a natural desire, I think, to have something that's your own and that you sort of lead. So when you develop a network, you're asking people to some extent abandon that and become more homogeneous. And there, to some extent, people consider that can be a bit stifling, and so there must be a delicate balance between establishing standards that are agreeable to everyone without stifling the creativity of people that feel like they need to do things a certain way, for example. Yeah. So
what are the benefits that you see when one decides to participate in this initiative?
Well, I would hope that there would be a common set of tools that would be available to them. I think any project, can't have enough resources to do everything well, so building off of other people's expertise, methods, collection methods, analysis methods. I think there's a huge benefit to being able to take advantage of that existing knowledge and communication, understanding what colleagues are doing in perhaps similar environments, what they're finding. I think anytime you have a network, it facilitates those kinds of interactions.
So coming now to the OSD, why do you think, is this a necessary initiative, and what are the benefits? Why is it important?
Ocean Sampling Day. Yeah, so, I was involved in the International Census of Marine Microbes, and we conducted a census of marine microbes, but there's still so much more that we don't know. I think it was something like, it was 1 divided by 10 to the 18th the volume of the ocean we estimated that we sampled. So despite everything we did, despite all of our efforts, which at the time were very important contributions, we've still sampled so little of most of the Earth, okay? And so I think, and also just to better understand how biodiversity changes over space and time, one needs to be out there making these observations. It's really important. And I think Ocean Sampling Day isn't just about microbial diversity, it's about all diversity. And I think moving towards all taxon inventories is really important because, as we know, there can be changes in communities that happen at different scales, and perhaps the microbes are somewhat sentinels to those changes in ways that higher organisms aren't, but will ultimately become impacted.
So following up on this change issue, do you think the OSD is also a good way to start monitoring about human impacts on the ecosystem?
Potentially, although I would like to see more long-term and continuous monitoring of environments. I mean, I think that's really critical. I mean, Ocean Sampling Day is designed to sample twice a year. I think we need to have in situ observations that are happening on a daily basis or even more frequently. So, and this technology exists. It does exist. It's in use. It's expensive, but I think there is the need and the desire, or there should be the desire. There is definitely the need, but there should be the desire and the resource to start to invest in this technology for that purpose.
So, in terms of maybe kind of low-hanging fruits, what do you think are the questions that in the near future can be answered with this initiative based on the data that you compile, this huge global scale consistent data set?
Right. So one of the things I'm doing with colleagues is looking at richness patterns across the global ocean, and that's based on data from the ICOM and also other projects. But one of the big questions is, how are diversity hotspots changing in our oceans? Where are the susceptible areas? Where are areas where pathogens are being born and bred? I mean, these are all types of questions I think that we're getting closer to being able to answer. And I think some of the work that Jack is doing in the EMP, thinking about how communities are changing and how that's reflective of the underlying oceanic changes. I mean, the ocean is intimately tied into microbial life, and better understanding the diversity and how it changes and why it changes is really important. And those are questions I think that we're poised to start to begin to address.
Absolutely. Mm-hmm. Yes, so I think, covered most of the questions. Is there something you would like to add?
I guess I just, in reflecting on some of the contributions the GSC have made as a network, as a team, and as a community, I'm happy with the progress that we've made. I think it's really valuable. It hasn't been easy at times to really motivate colleagues to be able to spend the effort and the time to keep track of data that maybe they don't feel is as valuable as I do. But nevertheless, I think we're starting to reap the benefits of that. And I just hope that we can keep up the momentum and that we can keep up the support, but also that funding agencies will recognize the value in funding projects and funding individuals that help keep this kind of activity alive, because it's not free. It does take resource, but there's a tremendous benefit and value added to it. So.
Right. That's a really nice final statement.
Thank you.
Thanks for your time, and thanks for answering all the questions patiently. Have a nice rest of the meeting.
Thanks.